So first, I will focus now on the biology and medicine part and the application of nuclear physics. So when we think about nuclear physics and medicine, we have to remember that this is a very long story. So as very little time after the discovery of radioactivity at the end of the 1800s, we got the first radiography by Röntgen in 1895, which was the radiography of the end of his wife using X-rays. And then, so of course, there was development on imaging and radiography, so improve safety and so on and so forth. But it was also until the end of the Second World War and the early 1950s, radioactivity was seen as good for health. So you were able to find commercial, for example, for public baths with pool of contaminated water with radioactive species, so generally radon and uranium and so on. So this was up to the 1950s. Of course, since then we have made some progress. So we know that radiation can have some benefits up to a certain level before starting to be detrimental. And we have done also some progress in the experimental nuclear physics part. So the detector that were used by Becquerel, Röntgen, and so on have been improved. So we are able to detect lower level of radiation. We are able to detect them, measure their energy with a better resolution, higher efficiency. But we are also now able to produce new isotopes. So the chart that I was showing you before has been extended in the last 20, 30 years quite substantially, both in the very heavy region here, then also in the very exotic neutron-rich region. So this implies some new techniques on how to select, identify the ions, and also to measure them. So if we focus more on the application of nuclear physics to diagnostics, so we think about imaging, for example, one of the first things that improvements made is in the detection resolution. So of course, if you get a better energy measurement of the radiation, you can improve the identification that you will get from the different radionuclides. So with lower statistics, you get still a good identification of which species is present. So here, for example, I show you a comparison between two spectra of the same source obtained with two different detectors. One that is detectors that have been used for quite some time, more than 50 years in nuclear physics, that is the sodium iodine, NaI, and a more recent one that is the lanthanum bromide, So what you see directly comparing the two spectra is that the peaks here in this region, close to 1,200 keV in energy, have a much better resolution with the new detector, that is the lanthanum bromide. So of course, having a source with only two peaks, one can say we don't really need this improvement. But this is a calibration source, so we know exactly what kind of radionuclide we expect, and we expect only one, that is, in this case, the decay of cobalt 60, so nickel 60. Then, but this will be quite important on the contrary if you have a source that has many transitions, or a source that is not coming from only one radionuclide but many, in which identifying the energy of the peak becomes more important. So here you have another spectrum obtained still combining sodium iodine and lanthanum bromide. on which you can see that peaks that before were very difficult to see with sodium iodine are now very visible and also also well separated. So this, of course, has some also implications. Since you have a better resolution, you need a lower statistic in order to identify the precision of the position of the peak and then determine the species. Then this, of course, after detection resolution, the second aspect is the detection efficiency. So you want to improve this aspect also. And so here it has been measured. So comparing two detectors that have exactly the same size, the data are taken in the same way, so same distance to the source and so on. And you see that the efficiency of the new detector is relatively higher than the one of the sodium iodine in all the different regions. So this, in terms of application to diagnostics, means that you will need to expose less the patient, for example, expose less the patient in order to get the same quality as the same quality for the imaging. Okay. So once you have detectors, what you need to do is to get a source of radiation in order to measure it and to do an imaging of the patient. So you need the development of radiopharmaceuticals species, and this can be done or using nuclear power plant reactors, okay, in which the fission of uranium in general will produce many species that can be extracted and selected. Or you need something with an accelerator. If you use an accelerator, you will need to have well-studied nuclear reaction, because you want reactions that have only large cross-section to produce your isotope of interest, because of course beam time is costly, so you don't want to spend more time than needed on this production, and you need reactions that are as selective as possible in order to avoid having contamination that can lead to false results in the diagnostic. So what can be done, for example, if you have beam nuclei, so the projectile that is sent on the target nucleus, these two nuclei will collide. Depending on how they collide, you can have fusion of the two. Okay, so you will do a nuclei that will rotate very quickly, and this nuclei can then or split into two, that is called the fast fission here after the formation of the compound, or start to emit charged particles or neutrons. So it will be neutrons, protons, alphas, so very light charged particles. And then you will get a final nuclei that is in an excited state. This kind of reaction that are called fusion evaporation reaction have been used in the past mostly to study this region of the nuclear chart. So all the region of the neutron deficient part. So the neutron deficient part. This as an issue is that this mechanism presents a big drawback for the production of radiopharmaceutical, is that the process of the evaporation of charged particle is a statistical process, which means that you don't have a correspondence one-to-one compound final nuclei, but you have many different final nuclei depending on the energy that you put into the system. You can have a few channels, 5, 6 different final nuclei, but you can also have 20 or more final nuclei, depending on the energy that you put in the system. So this kind of process is to be avoided when you want to produce radiopharmaceutical. So you need a different kind of reaction, and this can be done, for example, using transfer reaction. So in this case that I show here, you have a very light beam that can, here is a proton and a neutron, so a deuteron, that is sent on a target. In this case, the proton will be transferred to, from the deuteron to the target nuclei, and the neutron will be flying away. And you are interested in the nuclei B for radiopharmaceutical. So this kind of reaction is very selective, okay, and can be done, for example, in the AVION laboratories using the small cyclotron that is dedicated to proton beam and deuteron beam. So once you have produced your isotope, so for example, at the AVION laboratories, you will need to transport them into clean cells that are heavily shielded against radiation in order not to put the workers at risk, and in order to process them to be given to the patient. So generally what will happen is that the species that you produce will live for a short time, few hours, days, not much more, and will be fixed into a molecule. The molecule on which it will be associated depends on what you want to probe in the patient. So which kind of organs do you want to see, and so on and so on. So for example, if we take the fluor 18 here, molecules that are here, fluor 18 has a lifetime of 1 hour and 40 minutes. This means that the production site on which the fluor 18 is produced, treated chemically, and prepared for the patient, has to be close to the exploitation site, so to the hospital. Otherwise, you will not have enough material to be to do some reliable diagnostics. Another technique, for example, for technetium-99 that has a lifetime of 6 hours, is to knowing what is the all the decay that I was showing on the first map that we were having at the beginning. So we will look for molybdenum, for technetium. So molybdenum 99. So we look for molybdenum. So you see here that molybdenum 99, as I was saying, technetium 99, that is the isotope of interest, has a lifetime of 6 hours and 7 minutes. This kind of isotopes can be produced using accelerator base, but is not the most sweetened reaction, since the cross section and all the residues that you will produce make it a bit more complicated. So generally, this kind of isotopes is extracted from nuclear reactors. And we see here that is way more convenient to transport molybdenum 99, that has a lifetime of 65 hours, than to transport the technetium 99. Since you will have more time, you can make the production further away and transport your isotope. So this is for the production and the preparation. So once you have the isotope that is produced, you inject it into the patient. So using, of course, dedicated material for which the worker that is injecting will be protected. So you see here that the syringe is shielded against radiation. And then you put your patient inside the detector. So what will happen is that the fluorine will decay beta plus, emitting a positron. The positron, that is the antiparticle of the electron, so a particle that has exactly the same mass but opposite charge, will see an electron of the matter. The two will annihilate and emit two photons back to back of 511 keV. Detecting the coincidence between the two photons of 511 keV inside the PET scan, one can compute the position in which the positron annihilation happened. So you can determine with a very good precision where did this positron decay, which will give you information on where the process that you wanted to see. So for example, the tumor is located here in the brain of the patient. So once you have located the tumor, you want to treat it. And again, nuclear physics can be useful. So radiotherapy is something that has been used for quite some time now with X-rays or add-on. So the two have different advantages. If you use X-ray therapy, what you see here on the graph representing the dose deposited in the tissue versus the depth inside the patient, is that in the case of X-ray, you will burn a large dose will be deposited before the tumor, at the tumor, and then even at the in the healthy tissue that are after. In the case of hadrons that are protons and heavier nuclei, so carbon, oxygen, and so on, you will have a smaller deposit before the tumor, so in the healthy tissue before the tumor, and then very little deposit after the tumor. This is mostly because the mechanism in which the hadron and the X-ray are interacting with the matter is very different. So then, playing with the energy of the beam of these hadrons, you can scan the full tumor. So you can have the maximum energy deposit that is called the Bragg peak of the protons at different positions depending on the energy of the beam, and so scan the full tumor. So this is a different representation. So before sending the beam on the patient, you have to establish a planification of the treatment. So what is then used is different models on the energy, where you include the energy of the beam, X-rays or carbon ions, where and how to place the patient into the different for the irradiation. So in order to do this kind of planification of the treatment, nuclear physics, fundamental nuclear physics measurements are very important, because then again, you will need to know exactly how the carbon ions, in this case, will interact with the atoms of the human body. So as we are at quite large energy, so you will have fusion, evaporation, and this kind of process, but the two dominant process that you will have is the aberration. So you will have the two nuclei touching it, colliding between each other and forming what is called here a fireball. In the other one, ablation, you have quite a similar process with many more fragmentation, with many more residual nuclei. So all of these are inputs that are needed from nuclear physics, from fundamental nuclear physics, in order to compute all those cross sections, establish the planification, and so on and so forth. So here on this complicated graph, what is represented is the contribution of the different species to the dose when you do this kind of calculation using a proton beam, for example. So you see that most of the dose before the Bragg peak, this before the black curve, is coming from the proton beam itself. Once you have passed the Bragg peak, the contribution of the proton beam will drop because it has been stopped. And what you see is that the secondary particles, so the one that can be coming from scattering of elastic scattering or evaporation of protons, will start to contribute, start to be the dominant contribution to the dose deposited in the patient. So it is not true that in the case of hadron therapy, the dose stopped clearly after the Bragg peak. There is a tail that goes into the LC tissues, but this dose is much less than the one you will get for an X-ray treatment. So once the cell has been irradiated with the radiation, with the ionizing radiation, so protons, carbon, and so on, the DNA of the cell will be damaged, for example. If the damage are not so important, the cell can repair itself, or it will go to the process of cell destruction. So this can follow different paths and is more of the domain, of course, of biology.